pleasure to be sharing some of our data here today. My name is Christiano Alves, or Chris, if easier. So I'm an instructor at junior faculty in the Klein-Stiver lab here at the Massachusetts General Hospital and Harvard Medical School. And today I'll be sharing a bit about our work related to the development of new CRISPR technology to treat a severe disease, which is named Spinal Muscular Atrophy, or SMA for short. So the title of this presentation is a permanent genetic treatment for SMA using base editors. So just as a really high-level introduction, what do we know so far? We know that SMA is a very severe pediatric disease, a neuromuscular condition. I will provide more details later. And we know very well the pathophysiology of this disease. We also now know very well what's the genetic causes, what's the genetic mistakes that cause the disease. So the genetic mistakes underlining the phenotype. And thanks to the gene editing community, now we have tools, especially the ones I'll be talking about today, which are base editors I introduced later during the talk, that could potentially fix those mistakes. So the pitch here is pretty simple. Like, can we use these base editors to then change this genetic mistake, to fix the genetic mistake, and then improve the clinical outcomes in this disease? So the central question here, I learned to call this the MON test, which is basically a very simple way to explain what we're trying to do here, is can we use base editors as a genetic treatment for SMA? So down the road, can we keep developing these and actually provide a treatment, a permanent treatment for this disease? So let me break down this today in 5 main topics. So here's the agenda. So first of all, to make sure that everybody's on board, I will provide more background about SMA in both fronts, like in about the genetics and also about the clinical outcomes of the disease. Then we'll be talking a bit about the base editing and the development of these tools specifically to treat SMA. Then we'll show some data from our laboratory in terms of phenotypic recovery, so changing the phenotype of cells derived from SMA patients. Then I will explore a bit about the potential off-targets, potential side effects that we could have with this therapy, and how we are what we're developing in terms of gene editing technology to try to mitigate these effects. And finally, I hope to wrap up today with some in vivo data, so showing some data about the delivery of these base editors in a mouse model, an SMA mouse model. Let's first dive in the SMA background. All right, so SMA is primarily characterized by death of motor neurons. So those are the neurons in our body that innervate the skeletal muscle. So basically, these neurons are responsible for the innervation that controls all our voluntary movement. So when you have the death of those neurons, you have denervation. As a consequence, you have muscle wasting and muscle weakness. So the prevalence of this disease is about 1 into 11,000 live births. So this varies, depends on where you're checking, but worldwide we'll be talking about 1 to 11,000. And then in clinic, we can divide this condition in 3 subtypes. So SMA type 1, 2, and 3. So type 1 is the most severe of these types, and is also the most prevalent. So more than 50% of patients are usually diagnosed with SMA type 1. And just to be very clear here, to highlight, this is a very severe condition. So in type 1, we're expecting to have these babies, depending on the ventilatory special conditions, or the death by 2 years old. So it's a really, really very severe condition. The natural history of the disease is very severe. That said, what's causing SMA? What's the genetic basis of this disease? So basically, we have these loss of function mutations in a gene in our body, and the name of this gene is survival motor neuron 1, or SMN1. And then basically, when you think about like an affected subject, like a healthy person, we have 2 genes in our body, the SMN1 and the SMN2. And this is something that happened in evolution because most of the species, they don't have the SMN2. Just humans, mainly humans, have the SMN2 gene here. But basically, between these two genes, there are some, small difference polymorphisms, but one is very important, which is the C to T mutation in exon 7. So in the position 6 of the exon 7, instead of having a C, the SMN2 gene has actually a T. So when you think about like a healthy person, this is not a problem because that SMN1 is producing protein. But the SMN2 is not really able to produce 100% of full-length protein because of that mutation. So this is a silent mutation, so it doesn't change the amino acid, but changing very important binding site for a machinery that controls the splicing. So splicing is basically the process to making sure that the exons will be aligned in the correct order in the final mRNA product. So because of that T in the SMN2, many times the exon 7 is skip it. It's not included in the final RNA. So the bottom line here is when you think about a healthy, unaffected person, you have both genes producing protein, but SMN1 is producing a lot of functional and full-length protein. So not a problem. And then on top of that, we have the SMN2 gene producing some functional protein when the exon 7 is not included. I would say that that's about 10% of functional protein. When you think about a SMA subject, we don't have SMN1 gene producing protein. So because of mutations, it could be mutation. The most common ones are in the exon 7, but it could be mutations also in other locations. But the bottom line is that we don't have SMN protein, functional protein coming from the SMN1. So patients, that will be little, but patients can survive because they have the SMN2 gene producing some protein. As I said, really low functional protein levels coming from the SMN2 gene because of that C2T mutation. That's the genetic basis. So basically, we have mutations in the SMN1 that cause the disease, and we have this backup gene, SMN2, that can help to maintain some functional protein in the body. So I mentioned a lot about the natural history of the disease, but we also have to consider now we have finally some treatments. So we have so far 3 FDA-approved treatments for SMA. So one is under the Biogen umbrella, one's in the Novartis umbrella, and one's in the Roche umbrella. So in the next slide here, I will show some of the limitations, the principles and limitations of this disease, treatments. But what's very important to highlight is that none of these treatments are actually a permanent cure for the disease. All right, so here is just like a table that provides a summary, like a really whole picture about what's going on. So the first drug approved was the one that Biogen developed as nusinersen, or the commercial name is Spinraza. That's an antisense oligonucleotide that target that SMN2 gene that I mentioned before. But the problem with this type of therapy is that it requires a series of intrathecal injections. So it's a very complex delivery system. And also this targets mainly just the CNS. Then we have the gene therapy from Novartis, which is basically a gene replacement. So this therapy was approved in mid of 2019. And the idea here is to provide like a SMN1-like gene. Then we have some limitations with this therapy, including potential toxicity, and actually don't actually know the long-term longevity of this treatment, because we're talking here about a single dose, and we don't really know over the lifespan if the gene will be there producing protein. And finally, we have this small molecule from Roche, risdiplam, which it's also helping, especially if you think about a co-therapy scenario. But it's a small molecule administered every day, so patients have to take the pills. And I would say that the data is not as striking as gene therapy, for example. So conclusion of this slide, again, is that we don't have a permanent cure for the disease. So these therapies, just to be very clear, they transform the way that we see the disease. Before these therapies, as I said before, we were expecting to have type 1 patients dying with 2 years old or so, but now they live longer. So this is definitely transforming, but we have to keep developing better way to treat and potentially provide a permanent treatment. And then here's where we go back to the genetic basis. So just putting some numbers in that slide now. So if you have like a healthy person receiving 100% of the protein coming from the SMN1 gene and about 10% of SMN2 gene, you have a lot of functional SMN protein there. In an SMA patient, we don't have protein coming from the SMN1 gene, and it'll be very difficult to target those mutations because each patient can have a different mutation in the SMN1 gene. However, because the SMN2 gene is there, and we know that the C2T mutation is the main difference between both, the idea here would be to target that SMN2 gene and then make that T become back a C. And if we revert that T to C, we could activate the SMN2 gene to produce protein. How can we do that? So here's where the technology, the base editing technology, joins the game. So we can make, for example, A to G mutations using base editors. And if we target the other strand of the DNA, reverting an A to G, that will revert that T back to C. And that's what I'll be sharing in the coming section. So base editing for SMA. So first of all, let's just do a step back and recognize the genome editing revolution that we are living actually. Just to really point out, CRISPR, Cas9 system, and other variants, they definitely change the game here, especially because now we can design rational ways to edit the DNA. And this is I'd say the main thing here is that this is really cheap compared to technology that we had before CRISPR. When you think about nucleases, which were the first class of CRISPR that describe it to actually potentially do gene editing, we have to recognize that those nucleases, they cause double strand break in the DNA. So basically, by causing double strand break, we could have undesired products. So in this slide, I just summarize some type of double strand break caused by these nucleases, but thinking about the development of a therapy, that would be very complicated because that could cause insertions or deletions that we are not actually interested in seeing in patients, and we would not know the consequences. However, as the field evolve and grow, new technologies have been developed. And the base editors developed originally in David Liu lab, it is the one that I will focus here today, but also just want to point out that we have other technologies like prime editors and many other labs developing new technology to do precise insertions, deletions, replacements, talking about a single base to large replacement. So this is a really active field trying to develop new technology that could lead to new therapies. As I said, today I will focus on base editors, which was the technologies that I decided to pick to use for the SMA context. So when you talk about base editors, this has been growing as well. We have new technologies coming from the base editors, but in a very simple way, we could say that we have two main type of base editors: the cytosine base editors, or CBEs, or the adenosine base editors, the ABEs. And then ABEs is the one that we are more interested for this project because could make that C to A to G, and then that would be the C back to T. Okay. When you think about the SMN site, which is a site that we actually want to fix, in this slide, I'd like to focus attention in the target adenine highlighted in this slide. So when you see that in the position 6, so in red, we have that C6T. That's what we want to change. So if we target the complementary adenine in that nucleotide position and revert that to G, then we could make a complementary edit in the other strain, reverting the T back to C. So some challenges here, right? When you think about development technology, we actually have some critical challenges here. First of all, if you look at that neighborhood, we have other adenines that we don't want to edit. We call that bystander adenines, ACE. Why? Because we don't know what would happen if we added those, even if they are silent mutations, because it's a very tricky site where we have to control this splicing, where this machinery binds to control the splicing. We don't know what would happen if we added those other A's. So that's one challenge. The other one is this system, this CRISPR system, base editor system, to recognize this site and position the enzyme, the Cas9 or the Cas variant in this location. They need to recognize a pan site. That's a motif. Typically, we're talking about 3 nucleotides, and the canonical one, the white type one, would be a NGG. So if you look at the last one in this slide, so we're looking backwards, we have like GGA, so that would be a NGG, so any nucleotide and then GG. That's the only position that actually a Y-type enzyme could be positioned here, because if you look at the other options, every single other option there is not an NGG site, meaning that the Y-type enzyme could not be positioned there. So in a simple way to explain this is like it's really challenging to actually position the enzyme that we need in this specific location because of the limitation of PAN sites. So these are some types of challenge that we have when we're trying to develop base editor for a specific site, because we can't predict what's going on there around that target base editor. All right, so how can we overcome these limitations and develop something precise? So first of all, I want to share this information from Ben Kleinsteiber lab, the lab where I'm working on. This was published in 2020 in Science. And the idea here was to develop Cas9 variant that was not limited by the PAN site that recognized. So we call this SPRI, S-P-U-R-I, which is the last one in this slide. And then what you can see is that an enzyme that could recognize pretty much any location in the DNA. There is a trade-off there because also could produce more off-targets, and we'll talk about that later in the presentation. But for discover and exploratory research, that's a really great tool because you're not limiting yourself to NGGs in the genome. So we took advantage of we leveraged this SPRI Cas9 variant to see if we could do precise editing in that specific site, SMN2 site. So first here in this slide, in this plot, you can see the Y-type data. So the way that the data is displayed here, we title the region and then use different guides that would position in every single position that the edit window. And as you can see, the last one there, which is the guide A10, which means that the target adenine is in the position 10 of the window, is the only one that we can see some edit with Y-type. And we pretty much don't see across the other guides as expected because they are not NGG PANs. However, when we look at the SPRI data, which is this variant that I just point out, we do see a lot of editing across different guides. So this gives us the flexibility to use different positions and see which one is the best. So I'd like to highlight in this slide two main data. So if you look at the SPRI, which is the graph at the bottom, you have there the guide A7 and the guide A8, both are in the center of the slide. And you see that when you use this adenosine base editor 8E, which is a variant with some mutations that show that the deaminase component of these base editors are high efficient. So that will be the stronger green in those bar, the third bar. You see almost 60% on target editing with those guys, A7, A8. When you look at the guide A7, sometimes you see some bystander editing, so meaning editing in the other adenines that we're not interested. But when you look at the A8, we actually have a precise on-target editing with minimal bystander editing, if some, if any. Okay. So bottom line of this slide, using simple human cells, these are hack cells. So this is like our working horse here. So just to like have a model to develop these technologies, we can see that we can precisely edit like a single base that we are interested in that specific challenge site. Also, we compare these with other enzymes, for example, those developed from Davis Liu's lab. And also we see here using the guide 8 in this graph that SPRI still super high efficient, better than those other enzymes in this experiment. Okay, so I hope to convince you that you can now have tools. We develop tools that can precisely do that editing in human cells. But the question is, can we do this actually in the disease context, like in cells derived from patients? And that's the idea of this next topic. So to do that, we first collecting our hospital skin biopsies from 5 different patients. So those are all SMA patients, well characterized. And then they donate these skin biopsies so we can isolate the fibroblasts. So those are cells that would have the patient genome there, and we can grow that in cell culture and do transfections and edit those cells in the TC in cell culture. And as you can see here, to work with these cells, we have to sort those cells that are well transfected to make sure that we are exclusively looking at cells that we were able to deliver the base editors. And what we see is when we use that system, for example, the guide A8 with SPRI, which was the one that we pick as the most efficient one, we see a really, really high efficient in editing that target adenine. So what we see in this slide is data from 5 different cell lines, all done independently, and 3 of those you can reach almost 100% edit. So it's for sure over higher than 80% editing, and in some lines we reach like 99% on target editing. So it's a really, really efficient system to edit fibroblasts from these patients. And then the question is, okay, we provide on-target editing here, but what happens when we see that precise editing? Do we change something in these cells that could, mean something for the disease? And the first piece of data that I want to share is that the exon 7 mRNA expression, which is the one that's being skipped or not, it's actually really increased. And as a consequence, we also have more functional protein, like full-length SMN2 protein. So using this ELISA approach, we see almost a 10-fold increase there. So a lot of SMN2 protein being produced from the SMN2 gene. And just as a remind, these patients, they don't have SMN1 gene producing protein because they have mutations in the SMN1 gene. So what we see here, it's coming from the SMN2 gene. And then here, just complementary data showing that we can do this also with Western blots. And then we see some additional data in cells, like in other proteins like P10 that was previously described as associated with SMN. All right, so I hope to convince you so far that we can precisely edit that gene in that site, and we can precisely edit that site in the disease context in cell from patients. But now, when you do like that precise editing the window, we also want to take in consideration that this enzyme could recognize another site in the genome that would be off target, and that could cause some problems if they are, real off targets. So we dedicate some time to actually explore what would be the off targets in the SMA context. And this is experiment. This would be I need like a lot of time to explain, but basically the idea here is that you take the DNA from those fibroblasts, and then you do in vitro. You expose that DNA in vitro to the enzyme and the guide RNA, and you see what sites are nominated. So what are the sites that these enzymes can recognize? So in this graph, if you look, the green ones are the ones using SPRI, the gray ones using Y type. So you can appreciate that when you use the SPRI, which is the pan-LAS variant, you have more targets nominated because they are not limited by the pan site. So what we develop in the lab is actually this high-fidelity version of SPRI, which can bring those number of targets almost back to the Y-type levels, which will be the darker green in this slide. And these just nominating all targets, but here also a graph showing for the main targets, those that will get more than 1% of the reads, then showing that the shape of the graph is pretty much the same. So using Y-type, one problem is that we have a main off target, one of other problems that we are now learning. So if you see this graph, this is just data across like 5 different cell lines, and you see the star, the asterisk there, highlighting actually the on target one, and then you see like a peak, this in chromosome 7. That's, for example, a target that we don't want, right? That's a main off target using Y-type. But when you go to so this is using Y-type. When you go to the SPRI version, then what you see is that peak in the chromosome 5, that's our target site, the SMN2 gene. And then if you compare what you see on the first column, that's using SPRI, in the second column, that's using SPRI high-fidelity versions for each one of the cell lines. So the panels A to E, they are each one of the cell lines, the human cell lines. You see that using the high-fidelity versions, you just decrease overall this off-target editing. Okay, so here, like that might be a bit confusing because there's a lot of data, but here is a good way to summarize these in pie charts. And then we try to validate that, right? So we try to like see from those nominated ones, if you take the top 25% and check what's going on in cells, are they actually real off targets, or are we just nominated that in vitro and that's not happening in cells? And then here's the summary. So for the top 35, we will have the first one being SMN, so that's definitely added. So that's not included here. And then I'm showing the other 34, which would be the 34 next off targets. And what you see in fibroblasts is actually only 2 of those are real off targets in cells. So we actually statistically see some difference in cells. And in hex cells, because you are really saturating and expressing a lot of protein and guide, that might be we find more off targets. But then in the fibroblasts, which is the first pie chart, that's the one more in the disease context, and we see only 2 off targets. And actually, when you look the data, so the off target 19 in this slide and the off target 25 in this slide, those are the ones that are actually statistically different. You see like a small asterisk in the graph highlighting that. But we're talking about like something under 0.1% edit. So we're talking about like only 2 real off targets, and those are really, really minor edits. So under 0.1%. Okay, so this is just to describe how important you should look these off targets and how we can actually develop versions and high fidelity versions that could potentially mitigate that. And also just like a last piece of information here, when you look at this slide, the first one is the on target one, and you see that it reached almost 100% as expected. So we have a really high efficient on target and pretty much no off target. Cool. So yeah, so we keep developing that, and we keep like paying attention to potential off targets. That's something that our group does, and the field as a field we are trying to understand that, because if you have a main off target for your guide, you must replace that or find a way to mitigate that, because you don't want to learn about bad consequences of off target editing. So that's something that we definitely have to consider while developing therapies, gene editing therapies. So I hope to wrap up now with this last topic, which is basically in vivo editing. So can we actually deliver this in mice and see editing actually tissue? So this would be a complex system because, okay, it's not a human cell, but this would be more SMA models. And in the end, if you think about a therapy down the road, we need to deliver this in a live system, right? So first of all, we did these experiments using the hex cells again, just to understand if we could split the base esters in 2 pieces, N and C terminal. And the reason is because you deliver this in mice, these plasmids are so big, so you cannot fit these in a single AAV. So you need to split these in N and C terminal. So yeah, we did that, and we compare like traditional transfections in the lab, which you see on the left of this slide, with the one that we split in N-C terminal. And then just to highlight here, if you look, for example, the guide 8, which is the one in the center of this graph, we have like a really minor decrease in deficiency. So we still have like more than 40% on target editing using this dual system, this split of N and C terminal. And this is a very efficient system. So using this INTEIN and C terminal, after they're transfected, they fuse inside the cell and then make the edit. And as you can see here, is over 40%. So this is happening in cells. The question is, can you do this in mice? So to decide which mouse model to use for SMA, we took a look in the literature, what's available there, already developed. And we have some models that don't have the SMN human gene included, okay? So the 2 first models in this graph, the SMN delta 7, the Taiwanese models, they actually have a copy of the human SMN2 gene inserted in the genome. So those are the models that we could target, because everything that we develop is targeting the human SMN2 gene. So the third model that couldn't be used. And then we compare with these 2 models. The first one, the SMN delta 7, is actually the model that has a very clear phenotype in terms of motor neuron disease. So we decided to go for that one first. But as you can appreciate, this is a very severe model, as expected, because they're trying to mimic in a model what's actually happening in clinic. And it's a very severe and fast progression. Disease that progress very fast. So these mice, they would die before 15 days. So the window to edit here, it's really, really short. And okay, so that said, we tried to inject these animals in the first day of life. And then we did this in collaboration with Rashmi Guttari lab in Canada. And then basically inject this ICV, meaning that the distribution of the AV would be mainly in the CNS, in the central nervous system, and would reach the neurons. And then we look 13 days after, which is the time point that we can collect tissues from these mice. You will see that we get on-target editing, across different tissues. Because of the leakage, liver always will be edited. We will see this, over and over in different experiments. Well, what I like to highlight here is that we can see some edit in both brain and spinal cord. Those are the primary tissues for this disease. So in this first experiment, in a really short term, we can see a bit higher than 4% in spinal cord and around 6% in the whole brain. And this is not in sorted cells, this is in the entire tissue. So yeah, we need to learn later if this will be enough to affect the phenotype. We did also experiment here showing using mice that have the human SMN2 gene inserted, but not the SMA condition, meaning that the mouse model, the mouse gene is also there expressing. So these mice live longer, so we could do the experiment in a long term. So that would be 12 weeks or so. And then we can start to see more edit. So for example, in the brain, on average, now we talk about 10%, with some cases reaching 20%, and spinal cord about the same. So as we have a longer, like a larger window to see the edit, you start to see a better on-target edit. And as I said, our group now is working on phenotype related to these, production of protein related to these tissues. And then there are many other ideas that we could potentially use co-therapies or other types of the liver to see if we can make the base editors work faster. So that would potentially provide even better on-target editing. All right, so I'm running out of time, so I would like to summarize here what we know so far, what we learned from this data that I shared today, and then where we think that these things are going next, right? So SMA, a very severe disease. We know well the pathophysiology, and we have treatments in the market. None of those are permanent cures for the disease. So we know the genetic basis of the disease, and we know that the SMN2 gene is a good target. And then we now have base editors that could potentially edit that SMN2 gene and activate that gene to make that look like a SMN1 gene, and then potentially improve the phenotype and provide a permanent treatment for this disease. So I hope that I share enough data here to convince that we are towards a permanent genetic treatment for SMA. I'd like to invite you to take a look in our preprint. This work is under review. We are now working on experiments to complete this work. And then you can see this in the bioarchives if you are interested. If you want a more like a light, something more light to read about it, there are two nice pieces of news available in CRISPR News Medicine and Gene Edge. And those are interviews and also more details here. Both myself and Ben, we have Twitter, and we try to do a tweet tutorial there. So if you are into this type of like social media Twitter, you can take a look there. And yeah, so if you're more interested in this topic, those are really nice places to read more about it. And also I'd like to acknowledge here work that was just published a few weeks ago from David Liu lab on the same topic. So these were independent research done by independent groups, and then we are reaching very similar conclusions using slightly different components, but the conclusions, they are complementary. So in this paper, they have more mice data available. They don't have human cells, SMA patients, human cells available, but more mouse data available. So I think that these two works complement very well with each other. And then this is my last slide, just a bit about our group. And I'd like to thank Ben and the entire group involved in producing this amazing data. Shout out to Lani, which is down here, which is a technician in the lab about to graduate and go to medical school. And she really helped me a lot with this, all the funding and my collaborators. All right, thanks so much.